So I think the most promising checkpoint inhibitors that are currently studied, these are the most known ones like PD-1, CTLA-4 and TIM-3. And I think from perspective of the most important ones, I think that actually TIM-3 blockade is extremely promising, because we have seen that if you block checkpoint molecules like PD-1 or CTLA-4, you can drive graft-versus-host disease development in the context of allo-HCT. But we have seen that when you block TIM-3 specifically, we do not see an increased incidence of GVHD development. So we think that actually TIM-3 blockade is a very promising strategy, especially in the context of allo-HCT. I think so far actually there is not much clinical evidence for combination therapies of immune checkpoint inhibitors and other therapies, but I think that from the preclinical side there is a lot that we can learn actually. We know from our studies that we did that the expression of specific ligands are very important for the success of TIM-3 blockade, for example. So I think that when we find additional molecules that can block these different ligands that are expressed, I think this can really be a good combination strategy to enhance the graft-versus-leukemia activity. And also, what we're talking about is combining checkpoint inhibitors with allo-HCT, so I think this is the strongest immunotherapy that we have, so I think that's already a very, very promising strategy. Yeah, so I think it's actually combining immune checkpoint inhibitors and allo-HCT to kind of push the borders of allo-HCT and reduce the incidence of relapse after stem cell transplantation. And I think there's still more that we need to understand, like which kind of leukemias are actually responsive to these therapies, and is it, we have shown on the preclinical side that it's very important that we have the expression of the different ligands, and I think this now has to be proven in clinical context to understand which patients are really responding to these kind of therapies, and which kind of leukemias we can actually treat, and if it's really dependent on the oncogenic driver mutation, and if we can separate patients into responders and non-responders before we start the treatment.